Welcome everybody. I'm Lawton Snyder. I'm the CEO of the Eye and Ear Foundation. Our foundation, which has been in existence for 35 years, has always supported research in the departments of ophthalmology and otolaryngology. So our interest is in trying to help people with vision, hearing, and other ear, nose, and throat related conditions by trying to advance care for those who are part of the Eye and Ear Foundation spectrum. So this is the very first of our webinars that we've ever done. We've started this. We actually started this, of course, one, because we are at home right now, most of us working, but this is also an opportunity for us to find a different platform to try to get information out to you, which hopefully will be an easy way for you to get updated on the research we have going on at the University of Pittsburgh in these departments. I need to do a few housekeeping things with you first. And first, we are disabling the chat mechanism. If you're used to doing Zoom meetings, you usually do have a chat mechanism, which for now is not going to be the way we want to communicate with you. But you are welcome during the presentation to go to the question and answer bubble on the bottom of your screen. Click on there, and then you can type a question. We will hold the questions until after the presentation is finished, but then I will read the questions so that our panelists can answer those. We also are going to open up after the presentation the hand raising feature. The hand raising feature works like this. You click on the hand raise whenever you need to, and then I'll see that. And again, we'll call on you when you raise your hand, and we will unmute your microphone so you can ask the question. Again, that all will be done after the presentation. One last thing, as far as housekeeping, you will receive more emails from us. You're going to receive, after this presentation tomorrow, you'll receive an email that you can give us your feedback, your response survey on what you may want to have presented in the future from the research that we're going to talk about. But today you're going to receive, this presentation today is focused on vision. And what we're doing is an overview of all the vision research that's going on in the Department of Ophthalmology. And our presenter for today is Dr. Jeff Gross. Dr. Jeff Gross is the E. Ronald Salvetti Chair in Ophthalmology Research. He's the Vice Chairman of Research for the Department of Ophthalmology and Professor of Ophthalmology and Developmental Biology. He's also the Director of the Louis J. Fox Center for Vision Restoration. We've asked Dr. Vishal Janji, who is Professor and Chair and Cornea Specialist in the Department of Ophthalmology, to also be on the panel when we get to the question and answer section, so that you can ask questions that may relate more to some of the clinical research or clinical questions you may have. But the focus really is going to be on what research is going on in the department. Dr. Gross is the one who knows about all of that, and I'll turn the mic over to Dr. Gross to begin the program. So you should be able to see the slides in front of you that he's presenting, and you should be able to hear Dr. Gross now.
So hopefully everyone can hear me and see the slides. I'm going to, as I move around, I have a mouse here that hopefully I'll try to point to things and you'll be able to see it on the screen. So first off, thank you all for joining. This is the inaugural event, and I think this is an exciting way for us to try to get some of the research out to you while we're all working at home, or many of us are working at home. And with that said, I'll sort of apologize if my children jump into the room or my dog barks at something outside, where, this is the new norm for now. So this, I always like to start with kind of a general image, right? This is your eye. This is the thing that many of us are interested in. So it's just a cartoon of the human eye, and light will come into your eye through the front. This is called the anterior segment. So your cornea passes through the lens, which focuses it onto the very back of the eye where the retina is found. And this retina, in very simplified cartoon form, has a number of different layers. And at the very back are these photoreceptor cells. So these are the cells that the light essentially activates. And then those send electrical signals back in through these neurons to connect into the innermost cells of the retina, which are called the ganglion cells. And these ganglion cells then send axons out through what's called the optic nerve into the brain. So many disorders of vision can affect the front of the eye, can affect the cornea, can affect the lens. So cataract, for example. There are a number of degenerative conditions that affect these photoreceptors. So age-related macular degeneration, retinitis pigmentosa, diseases like this. And diseases affect these ganglion cells, the output cells of the eye, such as glaucoma. So we're interested in all of these tissues or all of these parts of the eye in the department. And as I go through kind of this broad overview of the research today, I'll try to highlight some of the teams we have that are working on each of these cells or in each of these areas. My next slide, though, is also an important one, and that's not to forget that we really see with our brains. So our eyes are very important to sort of bring this visual world into the brain, but our brain is where a lot of this processing happens. And this is kind of an exciting area in the department where we're growing, and I'll get to that in a few slides. But let's not always forget that our brain is important here. So I stole this slide from Dr. Sahel, our chair, and I think it's an important one because it really illustrates what we're doing and why it's important, right? Vision loss and visual impairment are increasing dramatically, and they're projected to continue to increase dramatically over the next 30 years. If you look at these graphs to the right side, this is vision impairment in red predicted, and this is blindness in blue. And up top here, hopefully this isn't too small, but you can see diseases like macular degeneration and glaucoma are really projected to both increase in number of cases over the year, and with age these increase. So as we're all living longer, these diseases are becoming more and more prevalent. And then on the left here, I find this kind of a fascinating study that across a number of ethnicities and demographics, people are asked to basically rank or indicate what they think the worst possible conditions could be. The choices were blindness, Alzheimer's disease, cancer, HIV, losing a limb, heart disease, arthritis, or deafness. And when you average out nationally, everyone that took this survey, people fear blindness the most. And that's because we're such visual creatures, right? We look at each other's faces for emotions. When you're having a conversation with someone, those sort of facial tics, the smiles, the look, seeing our visual world, this is really important to us. And people are quite scared of losing their vision. And this is what we're focusing on in the department, of finding ways to understand the visual system and then developing ways to hopefully cure some of these diseases. Advancing here, there we go. So the department, and this is something that really, it's been an exciting few years with Dr. Sahel taking over the chairmanship. We've been kind of thinking about ways to really focus the research from the patient to the clinic, right? And this slide, I think very nicely illustrates that we have a continuum that we're using to really generate innovation and then deliver that innovation to patients. So at the beginning of this, of course, are the patients. What are the clinical challenges? What are the conditions that people are coming in with to the clinic that require new solutions? This is coupled with a really amazing basic and translational science research program in the department, where the scientists work closely with clinicians to essentially take these challenges on, understand the root causes of the diseases, and start to develop targets and develop therapies for those. Those therapies, then we partner with industry, we partner with UPMC, we partner with a number of groups to further develop those such that they can get into some sort of a clinical trial and we can determine their effectiveness. This doesn't always work the first time, so this is the continuum. We then go back to the patient, back to the research, sort of optimize things, work with industry again, and so forth, until these new therapies are delivered to the patients. Within the department, the research can kind of broadly, very broadly be defined into the clinical and kind of the basic science realm. And I'll try to get into these a little bit throughout the next few slides, where we have expertise in imaging and artificial intelligence to interpret those images, clinical trials and restoration therapies on the clinical side, amongst many other things. And on the basic science side, really understanding how the visual system forms, how it functions, how diseases affect specific tissues or the eye as a whole, working towards discovering drugs and new therapeutics, and working towards developing regenerative and restorative therapies that can be then applied. The exciting thing is, I think this has been working really well for many years in the department, but what's particularly exciting is that, as many of you know, in 2022, we're still hoping that date is accurate, we're building this new hospital, the UPMC Vision and Rehabilitation Hospital, that would be attached to Mercy Hospital. And this is really exciting for us because these top 3 floors will all be research floors, just above all the clinical floors. So everyone will be in one place in one building, and really enabling this kind of stimulation of ideas and the back and forth between the clinicians and the researchers and the patients in one place. So we're quite excited about this. Moving ahead, I'm going to go through a few slides now to kind of talk again in broad strokes about the research in the department. And we're kind of divided into 6 or 7 different areas. So developmental and regenerative biology, genetics and genomics, visual information processing, modeling and imaging, and targeting different cell types in the eye. And I'll go through each of these now in the next few slides. So in the development and regenerative biology side, this area of research couples very nicely with the genetics and genomics. So really understanding how the eye forms, how specific cell types in the eye form, what they do, what happens in diseases to these cell types. We have a very strong focus on optic nerve regeneration that's funded through the Louis J. Fox Center. So understanding how, when the optic nerve is damaged, we can stimulate that to regenerate. And then this is coupled, again, very nicely with this genetic side, where we have a terrific clinician, Ken Nishal, who directs the pediatric ophthalmology side. We're trying to understand some of these congenital eye diseases and how they manifest, understand what happens to the cells when you have a particular mutation in the DNA of a patient. So maybe a cell type doesn't form correctly as a result of that mutation. So understanding all of that to really give us fundamental knowledge of the eye on which we can build to develop, again, some of these therapies. This is the brain side of things, the visual information processing, where we're really trying to understand what happens once that information is conveyed from the eye to the brain, how cognition and perception occur, and how this really feeds back into eye movements to understand the whole, sort of the whole system. This is exciting that many of you have probably heard through Lonnie and through some of the Eye and Ear Foundation mailings. We have a very large grant and support recently from the Richard King Mellon Foundation, which is driving growth in this area, that we really want to understand how the brain functions during vision. And this is particularly important for some of the restoration therapies. We think we're going to be able to sort of send information back or reactivate photoreceptors that might be damaged in the eye, but we need to understand how that information is sent to the brain, because there might be decades between when that sort of insult and when someone started to lose that vision and we want to restore it. So we really need to understand how the brain works in all of this. And that's an area we're growing quite rapidly over the next couple of years, and I think is quite exciting. Moving forward to think about disease modeling, and really this gets into pathways and therapies. I think one of the true strengths in the department, and this has been over the last 25 or 30 years, has been in infectious disease and immunology, understanding bacterial infections and viral infections of the eye. And this is largely stimulated through what we have is called the Charles Campbell Lab in the department. So we have researchers looking at immunology of the eye and what's called the ocular microbiome. So this is a popular term over the last, I would say, 5 to 6 years, where we have this system that's kind of in homeostasis. We have our own environment of bacteria and things in our bodies that really influences, it's thought to influence everything from disease to progression, from weight loss, things like this, really how all of this microbiome functions together. And we're lucky to have a terrific scientist that we hired a couple of years ago, Tony St. Ledger, who's directing kind of an immunology side of that and understanding the ocular surface and how some of these bacteria may be protective, actually, in keeping infections out. And then we have a large infectious disease group. As Lonnie said, Dr. Vishal Janji is here on the call, who will answer some questions later. So he's one of our corneal specialists. And then we have a number of faculty, Rob Shanks, Reed Kowalski, Kip Kinchington. Rob's a bacteriologist looking at the microbiology, so bacteria. Kip looks at shingles and virology, and Reed directs this Campbell lab. And this is a very exciting area because we're understanding these infections and developing new therapeutics to eliminate them. As far as pathways and biomarkers go, we recently recruited Dr. Debashish Sinha from Johns Hopkins University. He's the Jennifer Salvetti Davis Chair in research in the department. And Debashish is an expert in macular degeneration and aging, and really understanding at the back of the eye, we have these pigmented cells. And I didn't really call those out in the first cartoon I showed you, but they sit next to the photoreceptors in the eye, and they really help to support those photoreceptors. So in macular degeneration, those pigmented cells actually get sick and start to die, and that then influences the survival and the health of the photoreceptors. So Debashish is trying to understand the cell biology of this and really how these pigmented cells operate, how they sort of maintain their health and thereby maintain the photoreceptors. As far as therapies go, this is also, I think every area, I keep saying these are quite exciting areas. Every area is exciting in the department, but this is an area we've grown a lot also over the last 3 to 4 years since Dr. Sahel started. So first off, we have expertise in stem cell-based therapies. Largely this is for the cornea, but we're also, through the work of Yixin Du, we're getting into some glaucoma work where she's looking at how we might be able to restore some of the structures that are affected in glaucoma. As many of you know, we had a real giant in the field, Jim Funderburg, who passed away at the end of last year. And this is a huge loss to the department, to the community, and the field. That Jim's work on cornea regeneration is really, he's the star in this area. And the work he's developed over 30 or 40 years now, I think is quite promising, and it's in clinical trials in a number of different places. Luckily, we were able to recruit somebody named Gary Yam, who's also a cornea expert and regenerative biology expert, and he's taken over some of Jim's work with the help of Yixin Du, and they're really pioneering this work forward to keep it going and keep the legacy of Jim going and all he's worked on for that time. These two other areas are kind of intermingled very nicely, where we have experts in drug screening and drug delivery, Morgan Fedorchak and Yuanyuan Chen, and expert in gene therapy, Leah Byrne. And these groups are working together really to identify new drugs and compounds that might help to restore either the health of cells, to keep these cells alive in the eye, photoreceptors in particular, if they're affected in some of these diseases, make sure we get those drugs to the right place and in the right dosage, and that's Morgan's expertise. And then develop these new gene therapies, so viral vectors, that we might actually be able to put genes in or modify genes within these cells that are affected. So again, maybe within photoreceptors to restore their function, or to enable other cells to be able to respond to light and then hopefully restore vision in that way. And that's Leah Byrne's expertise. As far as phenotyping goes, this is a term that's really meant understanding all of the features of a disease, for example. So in the eye, basically, this involves in our department a lot of development of microscopy and imaging techniques, developing these new imaging systems, and then putting those in the clinic and really understanding this massive amount of data that's coming back. The idea behind this being we need to identify the disease as early as possible. If we're developing a lot of these therapies, of course we want to use them for people that have already lost vision or are losing vision, but we also want to use them as early as possible. So doing this deep phenotyping, really understanding what's going on at the cellular level, maybe in these pigmented cells during AMD progression, or in photoreceptors during some of the photoreceptor degenerative conditions, and in glaucoma. Can we start to really understand those phenotypes to predict disease and then use those predictions to target the right therapy to the right person? And we have a terrific team on the clinical imaging side and modeling side This is led by Ethan Rossi, kind of on the basic science side, and Kira Lathrop. And then we have two physicians, Kunal Dansangani and Jay Chablani, who were both recently recruited, that really focus on artificial intelligence and churning through this massive amount of imaging data to, again, be predictive and proactive so we can start to identify some of these disease processes early on. This is coupled to biomechanics and mathematical modeling expert Ian Siegel, who really understands how all of the tissues in the eye work together, right? It's not when you have a problem in one area, you have to fix that, but that kind of affects the system as the whole. So Ian really looks at this in a kind of holistic way and understands all of those processes. And again, predicts maybe ways that some of these therapies might help the eye. Coming back to this, again, this is the idea, this continuum, right? So a patient comes into the clinic with a particular disease or disorder, the clinicians identify that and they do what they can, but then we work together with basic science researchers identifying targets, taking this and working with industry partners and particularly partners at UPMC, putting this into some sort of trial and getting it back to the patients and doing this again and again and again until these innovations are really delivered in a way that hopefully solves some of these problems. With that, whoops, no, if I can go back. With that, I thank you for your attention. I know this is sort of a weird format to be doing something like this. I hate the term the new norm, but apparently I guess this is where we are. You know, we're grateful in the department for the unwavering support that many of you have had for the research and clinical activities here. And we're grateful for the attention you've sort of paid today to learn a little bit more about this. I'm happy to answer some questions. And again, I'm joined by my colleague and friend Vishal Janji, who's one of our physicians and cornea experts. So hopefully the two of us can help answer some questions you might have or stimulate some dialogue through this. So thank you for your attention.
Okay. At this point, I don't see any questions that were typed into the Q&A. If anybody would like to ask a question, please raise your hand. You see the raise hand button, and I'm going to look to see if anybody is raising their hand that we need to unmute during the session. So this is a very an overview of the research that is going on in the department, and thank you, Dr. Gross. It was a wonderful overview because there's a lot of information to cover. However, okay, I have a question here. Are you doing any work on retinas?
So I can start with that, and then maybe Vishal, if you want to talk about some of the clinical work, if it relates. But yeah, I would say about half of the research in the department is focused on the retina. We have researchers looking at specific diseases that affect the photoreceptors. So something like retinitis pigmentosa, where the photoreceptors degenerate and die. So we're trying to understand the biology of that. We're trying to develop or identify drugs that might slow down or prevent some of that, and develop gene therapy approaches to restore vision if someone has lost it because their photoreceptors have died. And then we have research going kind of through all parts of the retina. The ganglion cells, which generate the optic nerve, and those are affected in glaucoma. So we have people trying to understand how that disease process happens. Can we regenerate the optic nerve? Again, this is the focus of the Fox Center for Vision Restoration. There's an area of your retina called the fovea, which is what we use for high acuity vision. So if you're seeing me now or you're reading things on the screen, that's because of your fovea. So that's where you have this really dense accumulation of these photoreceptors. So we have a recent hire, Susanna De Silva, who's trying to understand how the fovea forms. And is that something that maybe through stem cell therapy, we might eventually be able to restore? So there's really retina research kind of on the basic science side from front to back of the retina. And I don't know on the clinical side, I don't know if you have anything you want to add, Vishal, if you know
Yes.
some of the clinical trials or things, please.
Yes, thanks, Chad. That was an excellent presentation. And, as you said, retina is one of our major forces of the department. It also has got something to do with the fact that our chairman is a retina specialist, and he's a world-renowned researcher. Um, there are a bunch of things, but I think the most exciting ones would be, one is adaptive imaging. Ethan Rossi has been doing that in close collaboration with the clinicians. And to make things simple, I would say clinicians have been so far able to see the retina through various examination techniques. We can image the retina to really, really near histopathological details. What Ethan is doing is combining the anatomical results with the functional results in this imaging. And some of the pictures Jeff had showed, Ethan has been building his own OCT platforms. In addition to that, the department is now already on the world map for running clinical trials that are based on gene therapy for certain diseases. Jeff has mentioned retinitis pigmentosa. There is Leber's congenital amaurosis and Stargardt's disease. Really, these are the neglected diseases where people go blind when they're as young as 30 to 40 years old, and they run in the families forever and ever. We've already started clinical trials on these gene therapy drugs. And the third, I would say most exciting, it was also on the news recently, is the development and implantation of a retinal implant, sort of a prosthesis which helps patients with severe blindness, which is secondary to age-related macular degeneration, which is the most common cause of visual impairment in adults who are age 65 or above worldwide. We did the first case recently, and the patient's doing really well. The way these implants have been, manufactured, Dr. Sahil has had a strong collaboration with the company. We work with them closely, and Yossi Martel, who's a retina clinician, he did the first case only about a couple of months ago, maybe a little longer than that. So, you know, in addition to that, there are a lot of clinical trials that are running. Dr. Herrera, who was recently recruited from Paris, she's been running a trial on a novel drug to control uveitis, which is intraocular inflammation that again is long-standing and is a major cause of blindness. But yes, as I said, implants and gene therapy and adaptive ocular imaging, these are three most exciting areas that we are working on.
Okay, we have a bunch of questions coming in now. Actually, this question I imagine for Dr. Jean G, of course, and this is something I know a lot of people are concerned about right now. Could you please explain how COVID-19 can be transmitted via the eye?
Okay, well, so we've had our Ioneer Foundation letter released, a newsletter released recently, Lonnie. I hope we can send it to all the participants as well. It sort of sums up what needs to be done and what needs to be avoided to sort of stop the transfer. I'm sure everyone has been reading the news. We have been doing that like crazy people now. These are, as Jeff said, unfortunately, these seem to be the new norms, at least for now. So not touching your face and not touching your eyes, that's top of the list. Washing your hands is another thing. I'm sure everyone's been doing that for 20 seconds. Everyone is doing it. Kids, adults, old, young, everyone's doing it. We wash our hands whenever we think about washing our hands, and which is a lot in a minute, I can tell you, especially when we are seeing patients in the clinic. Um, Specifically, I want to bring up one thing in terms of eyes, that you might have heard that COVID-19 infection can be associated with pink eye, which is conjunctivitis, and you know how common pink eye can be. And most commonly, pink eye is caused by another virus called the adenovirus. Um, there are very few reports where patients who had COVID-19 infection, they had red eyes. About less than 1%, I would say, about 0.89%, 0.89%. However, there is no report so far which says that a COVID-19 patient presented for the first time with pink eyes. That means there's no report where people have shown that conjunctivitis or pink eye can be the presenting sign for COVID-19 infection. So if someone gets red eyes, someone gets pink eyes and conjunctivitis, it's gonna behave in a similar fashion. So let's rewind 6 months. A patient comes into our clinic with pink eyes. We will tell the patient the usual stuff. Please don't touch your eyes. Please don't touch your face. If you touch your eyes, please make sure you wash your hands. Try and sleep in a separate room. Try and not share towels, linen, and pillows with anyone else. Try and make sure that your pets don't sleep in the same bed as you do. Please try and do this for 12 to 14 days. Now that sounds familiar, doesn't it? We're going to do the same thing if someone comes in with pink eye now. The chances of somebody presenting with conjunctivitis or pink eye with no other symptoms, no fever, no cough, no history of exposure to somebody who has an infection, no history of recent travel, the chances are that person does not have COVID-19 infection, only has pink eyes, has to follow the universal precautions, which are the same as we do with COVID-19. One additional thing, a lot has been said about contact lens use during these times. Now I know Dr. Dhaliwal, who's my division chief, she's in attendance as well, and she's a past president for the Eye and Contact Lens Association, and still a board member of the same association. Now we are not sure whether contact lenses can carry the infection. Not much research has been done. Obviously, the research now has been focused more on life-saving activities, not sight-saving activities in terms of COVID-19. But there is a possibility that if someone is a carrier for COVID-19 infection, they might transmit their virus onto a contact lens, and you don't want to leave these contact lenses out and about on your home shelves and stuff. The other thing is, if someone is a contact lens user, they would be touching their eyes and their face more than once a day, which kind of defeats the purpose. So we've been telling our patients, and it is everywhere out in the media, that if you're a contact lens user, please try and stay away from contact lenses unless it is absolutely necessary. Try and use glasses.
Or your
They give
glasses.
you, like Loan is doing,
Yeah.
they actually give you another layer of protection as well while you're using your glasses.
Okay, great. Now, this question, Jeff, you may want to answer because I think you did bring this up in the discussion. But explain more about statins in the use of glaucoma therapies.
So clinically, I'm not sure. I will probably have to defer to Dr. Janji for that. What I know and what I think is particularly exciting is, for optic nerve regeneration, one of our scientists in the Fox Center, Dr. Taka Kuwajima, has discovered that modifying statins to some extent really helps aid, and this is in animal models right now, so in mouse models, which is where Taka's working currently, aid in regeneration of the optic nerve. So there are a number of steps that will have to be taken before any of this could get into a clinical trial or into a patient. But it seems that statins and some of the molecular pathways through which they work actually have the capacity to help the optic nerve regenerate after it's been injured. And that's something Taka is now working with a colleague in the Department of Surgery, Dr. Steve Badalak, to sort of develop that project further with the help of the Fox Center. I know, Vishal, you probably could comment more on statins and glaucoma clinically. As I understand, it's a little uncertain.
It is. And I think you summed it up. There's a lack of clinical trials. I think the work on statins and glaucoma has been going on for nearly 8 to 10 years now. During my fellowship in the University of Melbourne, I think Ian Trounce and Crouchton was working on statins and glaucoma. A lot has been published, but there are no long-term results. When patients ask us, we ask them, "Are you on a statin?" So nowadays, there's a huge possibility that if someone's 50 plus, they are on a statin anyways. So it's kind of, if it has a protective effect, we tell them, well, you're covered. We don't know what is going to happen. But of course, for now, there's a lack of randomized clinical trials. So we really don't know if there's a perfect answer to that.
Right. The next question here actually is from Dr. Sundaraj. Good to see Dr. Sundaraj. His question actually is for Dr. Vishal regarding any work with artificial intelligence for dry eye.
Okay. Hi, Sandra. Good to see you. Good to hear from you, I guess. Um, so yes, there are certain groups that are working on AI in dry eyes. Um, it's it's an underutilized, side of medicine, especially in dry eyes. We have been working on dry eyes, not on AI though. I'm sure you are aware of what is happening in the clinic. Again, Dr. Daliwal and myself and other researchers in the cornea division, they are associated with dry eye and imaging as well. Most of the artificial intelligence work in dry eye is associated with these machine modeling techniques where we are able to pick up patients with dry eye, especially when they are on treatment for dry eyes. So most of the AI in dry eye has been used to actually evaluate the efficacy of different treatment modalities in dry eye. Nothing much has been done beyond that. But of course, in terms of other ocular surface diseases, artificial intelligence is moving forward pretty fast, like keratoconus, pterygium. These are things that affect the ocular surface.
Okay, thank you. The last question on the board right now actually is from my good friend Melody Goodspeed from down in Virginia. Melody's question just disappeared. Hold on. Melody's question was related to optic nerve regeneration, but it just disappeared on me before I got a chance to read it. Let me see if I can read the answers. Wait a minute. Um, no, it just disappeared on me. So
This is Jared with the Eye and Ear Foundation. I can read it. I can see it for some reason. The question is it common for other parts of the eye to deteriorate once the optic nerve has experienced damage?
So I think, and again, maybe Vishal and I can tag team this one from a clinical and a research side. One of the things that is really affected when there's optic nerve damage is the ganglion cells. So again, the ganglion cells are the innermost cells of the retina. If you remember that cartoon I showed at the beginning, the photoreceptors are at the back, the ganglion cells are at the front. So when the optic nerve is injured or damaged, those ganglion cells die. And this can be from neuropathies, from ischemia, from glaucoma, all different sorts of things. So those cells are lost. And it's often a pretty rapid process, at least in experimental models, when you create some sort of axonal damage to the optic nerve. Beyond that, I don't know how it affects the connectivity, which would be the subsequent connections out towards the photoreceptors. Visual, you may know from looking at clinical images, but I think things tend to be intact relatively, at least in the experimental models. So please maybe jump in if that's not the case.
Well, no, we're nearly there with the answer. I think that's a very interesting question. And the answer really depends on the cause of optic neuropathy. Let's take two examples. Let's talk about a patient who had a massive trauma to the eye, and the trauma actually only resulted in optic nerve damage and nothing else. So in a patient like this, there would be profound vision loss. The patient would not be able to see anything out of their eye. But the rest of the eye actually will stay intact. Okay, the front of the eye will look perfectly fine. The eye will look like an eye from a distance. However, that might not happen in real life that often, fortunately. Glaucoma is one of the most common causes, high pressure, for optic neuropathy. And typically patients who have glaucoma, while we are trying to slow down the death of the ganglion cells, that's all we can do for now using eye drops and surgery to lower the pressure. We do see very commonly that these patients will come back because of the side effects of those eye drops per se, and the side effects from the surgeries that are essential to bring the pressure down. So Jeff's answer is perfect in a way that if you think about optic neuropathy and optic nerve damage, individually, it is not supposed to cause any harm to the other structures in the eye. But when you take the eye as a whole, and look at the cause of the optic neuropathy, which most commonly is glaucoma, the patients might suffer from other things because they are being treated for that optic neuropathy. I hope that answers your question to a certain extent.
Great. And so then I've got a question, and actually a few questions here from Dr. Ron Salvetti. Hello, Dr. Salvetti. Um, uh, Dr. Salvetti's question is, what research approaches seem most promising in discovering a treatment for macular degeneration? And then he sort of gave us some specifics: stem cell, gene therapy, or other.
So this is, hi Ron, this is, as you know, an extremely active area of research, certainly in the department with Debashish Sinha's lab, my lab, a number of other labs. You know, I think everyone's taking kind of a multi-pronged approach. So stem cell-based approaches hold promise. You know, there are a number of trials right now that are getting started using sheets of cells. Remember, again, at the back of the eye, you have these pigmented cells that sit next to the photoreceptors and keep those photoreceptors happy. So when these pigmented cells get sick and die, the photoreceptors then also start to die. So there are a number of clinical trials underway where sheets of stem cell generated pigmented cells, these are called the retinal pigmented epithelium, sheets are being put into patients, or at least being developed to be able to put into patients to see whether those might be able to keep some of the photoreceptors happy. So that's one set of approaches. People are looking for compounds and drugs, and this is something that Debashish Sinha in our lab, or our department is also working on, to basically find compounds that might help these pigmented cells do their job a little bit better as they get older and they start to slow down, or they're being taxed by all of the photoreceptors they're taking care of. Are there compounds that perhaps could be delivered to help rejuvenate them a little bit, or help get rid of material that they're not able to degrade and eliminate anymore, which is part of their normal job? So I think on the drug and pharmacology side, there are some promising things happening. And then there's also a somewhat more recent area of research looking at the immune system and inflammation as it relates to macular degeneration. And this is something that Dr. Sahel also still runs the Vision Institute in Paris, and there's a kind of a world expert in this area, Florian Senlab, who is one of the researchers in Paris. And he now is working a bit with Debashish, a bit with me, looking at this inflammatory response. And in our case, we're trying to see whether we can stimulate these pigmented cells to regenerate on their own, which I think is a promising approach as well. Requires more research, but can we get these cells to start to divide naturally, sort of jumpstart this ability to regenerate a little bit? And it seems like the immune system may play a role there in allowing them to do so. That inflammation can sort of be good and it can sort of be bad depending on the context. And Debashish, I believe, is looking at the inflammatory response, and if that can also be controlled a little bit, because as you start to get buildup of all this material in these or outside of these pigmented cells that they're supposed to be getting rid of, that induces the immune system to sort of attack. And when the immune system tries to get rid of this material, which is its job, in doing so, it actually makes these pigmented cells a little bit sicker. So maybe if we could start to control this immune response a little bit, we could keep the cells alive longer, which would then allow people to see a little bit longer with AMD while some of these more restorative therapies are being developed. So I think I've almost covered all of the options, in a sense that you have stem cell approaches, you have gene therapy approaches, you have drug-based approaches, either targeting the pigmented cells themselves or targeting the immune system. And I think a lot of these are promising. There may not be one silver bullet that one approach works for every form or in every patient, but I think if we continue to develop all of these approaches, maybe for some patients a regenerative approach would be really appropriate. Maybe for some, controlling inflammation or helping the aged cell rejuvenate a little bit might be another approach, whereas for others, a stem cell or a sheet-based therapy might be the best approach. So I think they all hold promise. They all still have questions, and that's what the researchers are really diving into now to try to solve. So hopefully, within that ramble, I answered your question.
That was very, very well done. So one more question here, and it's from Leah. She's asking if there's any work being done for autoimmune retinopathy or AZOR. I'm hoping that maybe Dr. Jean-G may be able to chime in as well.
All right. Well, the short answer is no. We are not doing much, and one of the reasons is that fortunately or unfortunately, we don't see many cases of AZOR. It's a disease that was described mainly from the Far East, originally from Japan and China as well. As I mentioned, we don't see many cases, but I can tell you two things that have not been resolved in AZOR, and we might have answers to that in near future, is the cause for AZOR, what happens in AZOR, because that'll help us to understand the role of new immunomodulatory drugs in conditions such as AZOR. And I can tell you the number of immunomodulatory drugs that were available systemically and now are available for ophthalmological use, it has increased, by many folds. In ophthalmology, we are now using drugs to treat diseases like AZOR and other diseases which have sort of a dysfunctional immune system related causes. It is just tremendous. And thanks to our rheumatology colleagues as well. The second thing that we have really developed is imaging. Marie Ellen Herrera, I mentioned her a couple of times. She is an expert on ERG, electroretinography, and also we have access to multifocal ERG. And Dr. Shah, who was recruited from Johns Hopkins, he's an expert on that as well. But yes, as I said, we do understand for any disease that we have to work on it, we have to see patients. If not, we at least need to have an animal model. And because of the rarity of the disease, and I'm not trying to undermine the importance of the disease, it has been kind of difficult to, do more research on conditions like AZOR. But who knows? At least we are well armed now.
Thank you very much. That looks like all of the questions. I think everybody chose to do the typed questions. I didn't see any hands being raised, and I certainly hope that wasn't because people were trying and we had some difficulty in seeing that. But it looks like that's all the questions. I want to first thank the panelists for your time and ability to answer these everybody's questions today, and thank you, Dr. Gross, for the presentation, the overview. As I said earlier, we are planning to repeat these. We will cover other topics. We have plans covering topics for both of the departments that we work with, so both the ear, nose, and throat and vision care side of what Eye and Ear Foundation supports. So look for future webinars through what you receive from us from the Eye and Ear email blast that we send out. Now, I noticed that there were some new people on this list. I also noticed that there are some, I do know that many of you have friends that have interest in some of the work that's going on. So I'm going to share with you right now the link to sign up for our newsletter, our online newsletter. That link is eyeandear.org. So altogether, eyeandear.org/newsletter/. And then you'll be able to sign yourself up to receive our newsletter and information on things like this with our webinar. So you can share that link with any of your friends. Okay, thank you very much. We look forward to getting feedback, which we will send out again an email survey to everybody on today's call. And I'm not seeing any other questions, folks. So I think we are going to end today's webinar, and we really appreciated to have a chance to talk to you. I'd much rather do this in person, but if we during this time. It does give us an opportunity to do what we love to do, which is share the really good work that's going on in the departments, and how much we appreciate the fact that we're inching closer to solving some of these problems that really affect our patients. So thank you all again, and we'll look forward to seeing you next time.
Thank you.
Thank you for your attention.